and both my lab and I think this department as a whole are much the better for it. So I honestly think I could talk about Matthew for the full hour, but I don't want to cut into his time too much, so I'm just going to emphasize three things that I've really come to love about Matthew. The first is the breadth of his intellectual curiosity, both within science as well as extending to philosophy and the humanities and really all other aspects of life, and his willingness to engage in really fun discussions about pretty much anything. And I think this is something that is increasingly rare, and just has been great to have as a part of the lab. The second is scientific, his creativity and rigor when it comes to the work he has done, and I'm sure the work he will do. I'll let him talk about the work himself, but you know, Matthew, it's easy to forget, came in here with virtually no wet lab experience, was extraordinarily interested in diagnostics, I think, following on his experiences in Africa, and developed an expertise in an area around cell-free DNA, and proceeded to both conceive and execute on I think, some really brilliant ideas in this space. You know, I was once told that when you finish your PhD, there should be one thing that you know more about than anyone in the world, and I think for Matthew, at this particular moment, that really is the case. Last and certainly not least, I'd like to acknowledge what an integral and positive member of our community Matthew has been, and I'm really talking about not just my lab here, but the department as a whole. There are, you know, I can point to some concrete things. For example, organizing courses like the philosophy of genomics that Maynard and I think Bob participated in teaching. The Dick Lewontin Journal Club, a very specialized journal club for Dick Lewontin aficionados. And stepping up effectively to serve as faculty at one point for teaching statistical genetics to his fellow graduate students. But I think as important as any of those more concrete things has been kind of the more under the radar, consistent and proactive role he's played in contributing to this and helping to maintain this remarkable community that is this department over these past years. So yeah, I really could go on for an hour. Matthew was really like I was trying to think of an analogy, and the only things I could come up with were an onion, many layers. Thomas Pynchon novel. But I'm going to shut up and let him talk. So just to explain the order of events, Matthew will have till the end of the hour to present his seminar, and then we'll take some questions from the audience, and then everyone is excused except for his thesis committee, which will stick around to ask him some hard questions. And there are food and drinks somewhere. Does anyone know where?
Third floor lounge.
Third floor lounge. And Matthew and the committee will join you up there as soon as we can. So, okay, Matthew. Switch over here.
Yeah. Give me 1 second to recombobulate myself here. Time to let that moment sink in. Check. Here we go. Is that volume okay for everyone? All right. One last thing. Great. Well, it's really great to see everyone here today. I'm very excited and a little nervous, honestly, to be up here sharing my experiences over the past 5 years, and hopefully I can convince you that some of the work I've done is meaningful and will have clinical relevance. And specifically, I want to talk to you about the work I've done at looking at cell-free DNA, as Jay pointed out, so kindly in this very lengthy and elegant introduction to me, where we've really tried to expand the accuracy, the resolution, and also the breadth of the things that we can look at with cell-free DNA. The search for biomarkers of disease is really as old as medicine itself. The art of uroscopy, or the examination of patient urine samples to learn about diseases that someone might have, dates back well over 1,000 years at this point. And here I'm showing a so-called urine wheel, where the colors of urine from a patient sample were matched to this wheel in order to diagnose a patient. Now, the urine wheel might seem a little bit quaint, and the diagnostic utility, it wasn't perfect, but nonetheless it did actually have some success. For example, kidney disease was evidenced by a shift towards a dark brown color of the urine. And color wasn't really the only feature that proved useful. In fact, in the 1650s, Thomas Willis discovered that in individuals with diabetes mellitus, the urine had a pronounced a sweet taste owing to the glucose in the urine. Slightly later, Frederick Deckers discovered that in patients with proteinuria, the urine sample from that patient, when boiled with acetic acid, would yield a visible precipitate. So while medical technology has certainly improved in the 500 years since then, in some ways there remains this striking link to the past, as we can appreciate by looking at this colorimetric, this color change in a dipstick, which is still conventionally used to diagnose proteinuria. Biomarkers in blood have had a similarly long and checkered history, not always with the greatest success. Galen, who was a physician in ancient Rome, believed that diseases were caused by an imbalance in the four humors. By taking a sample of blood from a patient, he would wait for the blood to settle, and then essentially look at the different proportions of the four humors, what we would now call plasma, white blood cells, and different types of red blood cells. For example, jaundice was marked by an increase in the proportion of yellow bile. So while a large number of new and powerful biomarkers have been discovered in the blood since this time, nonetheless there remains a striking link to this past. The erythrocyte sedimentation rate, or ESR, shown here at the top, is a relatively common test for inflammation, performed by measuring the rate at which red blood cells settle during the course of an hour. Hematocrit, or the percentage of red blood cells in the blood, is an important diagnostic tool for anemia. So while diagnostic accuracy has certainly improved since these ancient times, both of these tests actually bear a remarkable similarity to early tests devised before the Cartesian revolution in biology sort of revolutionized the search for new biomarker discovery. And what a revolution this has really been. Today, an annual checkup at the doctor would likely result in what have been standard, would likely result in what are now sort of standard panels of blood tests, CBCs, a metabolic panel, various enzymatic tests, blood chemistry. Now, these tests provide a lot more information than we can get just by looking at a tube of blood, and collectively they've had an enormous impact on health, both at the individual level, for example, in the context of detecting infections or heart attacks, but also at the social level, helping to guide, for example, federal dietary recommendations. Nevertheless, these biomarkers are still somewhat crude. The genomic era of the past 20 or so years has ushered in a push for biomarkers of precision medicine using these high-resolution biomarkers. So what goals might these biomarkers help us achieve? One of the key aims of precision medicine is understanding not just what broad category of disease an individual might have, but what specific features of that disease might be useful to design a customized treatment plan. Biomarkers are also useful after a diagnosis has been made, both for monitoring the burden of a disease and for detecting potential recurrence of that disease down the road after treatment. Let's look at two examples. The Philadelphia chromosome is a feature of many chronic myeloid leukemia patients in which sections of chromosome 9 and 22 are essentially swapped, creating a fusion gene called BCR-ABL, which is a hyperactive tyrosine kinase. The presence of this fusion is not only a sensitive test for CML, but also predicts the response to Gleevec. Similarly, about 20 to 30% of breast cancers are what's called HER2 positive, marked by overexpression of HER2, the cell growth factor receptor. Individuals with HER2 positive breast cancer may benefit from treatment with Herceptin. So these are undoubtedly useful tools for many patients, and are certainly compatible with this modern hypothesis that our genetic makeup explains at least a substantial portion of our disease risk. These are both for serious congenital disorders, as well as for complex traits, including lipid levels and macular degeneration. However, many of these emerging biomarkers require the use of invasive and uncomfortable procedures, like tissue biopsies, for example, and inpatient procedures in general. Ideally, what we'd like to find is a biomarker that's powerful, informative, non-invasive, and compatible with today's standard clinical workflows. Today, I'm going to talk to you about one such biomarker, cell-free DNA, which I've spent the bulk of my PhD working on. I'm going to try to convince you of the enormous potential of cell-free DNA in various different contexts. I just want to emphasize the word potential here, because while the stories I'm going to tell you today, I think, are promising, there's still much work to be done. Let me just briefly introduce the history of cell-free DNA. Almost 70 years ago, Paul Mandel and Pierre Mattei unexpectedly found nucleic acids, in this case both DNA and RNA, in the plasma of various individuals, both healthy individuals as well as a seemingly randomly assembled cohort of individuals with diseases ranging from tuberculosis to gout to obesity. What they were observing was a large number of molecules of DNA outside of cells, or cell-free DNA, which they could purify from the plasma of these individuals. These molecules represent the detritus, or the debris of cell death, the bits of the chromosomes that are produced when cells die and undergo apoptosis, and their components are broken down for recycling or excretion. This observation went, roughly speaking, nowhere. It was essentially a historical curiosity until the mid-80s, at which point there was a surprising discovery that in some individuals with cancer, a portion of that cell-free DNA appeared to derive from the tumor itself. The next decade, Dennis Lowe found evidence of fetal DNA, or actually what we now know more precisely to be placental DNA, in the plasma of pregnant women. These observations showed that different tissues, different organs were able to contribute to this pool of cell-free DNA within a single individual, suggesting that perhaps it could be useful for diagnostic purposes. So by this time it was clear that cell-free DNA had diagnostic promise, although I'm not sure that even at that time the researchers quite appreciated the degree to which cell-free DNA would take off. Over the next decade, three key applications of cell-free DNA to clinical questions emerged. In particular, we can now use cell-free DNA to evaluate the health of a fetus during pregnancy, to detect or to guide the treatment of an increasing number of cancers, and to monitor the health of transplanted organs. Improvements to genomic technologies that are needed to interrogate cell-free DNA argue that I think additional clinical applications are on the horizon, although we're not quite there yet. Now, the genomics field has taken some admittedly deserved criticism in the past for over-promising. And we're clearly not immune to this even today. Recently, two prominent geneticists published an article in which they argued that cell-free DNA might actually be a stethoscope for the next 200 years. Well, I would suggest that the stethoscope is likely to be the stethoscope for the next 200 years. Nevertheless, I think it's clear that we've only scratched the surface with what we can do with cell-free DNA. And today I'll tell you three stories about how we might be able to improve the degree to which we can investigate various conditions with cell-free DNA. In particular, I'm going to tell you about approaches I've developed to improve the accuracy, the resolution, and the breadth of cell-free DNA investigation. And I'll start by telling you about how we can improve the accuracy in the context of pregnancy. Many women find themselves in a prenatal care setting, at least once during their lives. For some women, due to advanced maternal age or previous pregnancy outcomes, this can be a stressful experience, as they may be recommended for prenatal testing for serious fetal conditions, such as trisomy. Trisomy is a condition in which an individual has an extra copy of a chromosome, 3 copies instead of the typical 2. The 3 most common trisomies are known as Down, Edwards, and Patau syndromes, and collectively affect approximately 1 in 700 live births in the US. A series of increasingly refined and accurate screens have been developed over the years to identify affected pregnancies, including sonographic and protein-based screens. However, these screens still fall short of a diagnostic threshold, and so confirmatory procedures are needed in the event of a positive screen. While these diagnostic tests represent true gold standards, they nonetheless carry a small risk to both the fetus and potentially to the mother, and are certainly, even if not risky, quite uncomfortable procedures. And these include amniocentesis, chorionic villus sampling. So a non-invasive screening method that could achieve very high accuracy could hopefully reduce the burden of invasive follow-up